During the progression of most cancers, tumor cells must find ways to evade destruction by the host's immune system. One such biochemical mechanism recently identified with evasion of T-cell destruction in many human cancers is the PD-1-PD-L1 receptor-ligand interaction. Programmed cell death receptor 1 is a receptor expressed by T cells, B cells, and monocytes in the human immune system that mediates various immunological response pathways. The whole PD-1 receptor is a 268 amino acid monomeric type 1 surface glycoprotein consisting of a single V-set immunoglobulin superfamily domain attached to a transmembrane domain and a cytoplasmic domain, with two tyrosine-based signaling motifs. PD-L1 is a ligand both constitutively and inducibly expressed on antigen-presenting and tumor cells. PD-L1 is believed to interact with PD-1 in a highly localized region of the receptor. NMR studies have shown that the formation of the PD-1-PD-L1 complex is followed by perturbations in the PD-1 backbone, which include, but are not limited to, alteration of glutamine residues centered along beta sheet residues. Additionally, 33 other residues have been shown to be perturbed during the PD-1-PD-L1 ligand interaction. Of significance is the fact that these form part of an interaction surface. This has great implications for the mechanism of action, which we will now discuss.
When PD-1 cross-links with its ligand, PD-L1, the immunoreceptor tyrosine-based switch motif on the cytoplasmic end of PD-1 becomes phosphorylated. ITSM then recruits phosphatases SHP1 and SHP2 to act on another cellular pathway, the T-cell antigen receptor pathway. SHP1 and SHP2 act on the ZAP70 signaling kinase of the TCR pathway and dephosphorylate it. This interferes with the TCR signal transduction and ultimately places the T-cell cycle in arrest between the G0 and G1 phases, but does not elicit cell death. Ultimately, this interference hampers the T-cell's ability to conduct its normal functions of proliferating, secreting cytokines, and killing infected cells. These effects become exacerbated by the fact that the PD-1-PD-L1 axis operates primarily during chronic infection. In the presence of antigens, both PD-1 and PD-L1 become upregulated. The relevance of the PD-1-PD-L1 interaction during chronic disease uncovers its evolutionary significance. This pathway evolved to regulate immune-mediated damage during persistent infections by effectively interfering with the TCR pathway and turning off T cells. Indeed, studies show that in facing a chronic infection, PD-1 deficient mice experience a 0% survival rate due to immunopathologic damage, while the wild-type mice experience 100% survival. Fortunately, since the PD-1-PD-L1 interaction places the T cells in a state of arrest rather than eliciting an apoptosis pathway, the damage done to the T cells is indeed reversible by providing anti-PD-L1 treatment. In one study, mice were infected with the lymphatic choriomeningitis virus. Upon anti-PD-L1 treatment, the treated mice displayed significantly reduced viral counts across various organs as compared to the untreated mice. This can be attributed to the resurgence of immune response.
Due to its role in immune evasion, renal cancer cells have been demonstrated to overexpress PD-L1. Accordingly, disrupting the PD-L1-PD-1 interaction may render cancer cells susceptible to immune destruction. Using this as a framework, various groups have designed high-affinity monoclonal immunoglobulin antibodies to disrupt the PD-1-PD-L1 interaction. Specifically, MPDL3280A was designed by screening a human phage library against the extracellular domain of human PD-L1. Affinity measurements revealed MPDL328A to have a dissociation constant of approximately 0.4 nanomolar. The antibody was judged to block the extracellular domain of soluble PD-L1 from interacting with PD-1. The designed antibody was shown to increase T-cell activity as well as decrease tumor size in a number of patients. Additionally, genes associated with T-cell activity were significantly increased compared to their pretreatment levels. Due to promising results of MPDL3280A, many antibodies attempting to disrupt PD-1-PD-L1 are currently in development. These antibodies target differing regions of either PD-1 or PD-L1, but all operate under the assumption that disruption of the interaction will yield therapeutic benefit.